Thanks so much. Our next speaker this afternoon is Jennifer Cruz Sanders, who is the director of the State Botanical Garden of Georgia, which serves communities in Georgia and beyond with educational programming, horticultural expertise, and display gardens. Jennifer has worked to build networks for conservation across the Southeast and to develop sustainability programs and establish native habitats. Today, she'll present from dwarf sumac, Rhus mashoi to turkeybeard, Xerophyllum asphodaloides, and other species in between, how conservation horticulture has helped to preserve imperiled species in the southeastern U.S. Technical difficulties. The mouse's trackpad does not function. You have a magic finger? Oh, goodness. What did I do wrong? Help. Yeah, I see that. Yeah. There we go. Okay. There we go. That's looking better. Okay. There we got it.
Good job.
Just in time. Thanks.
Good afternoon. It is an honor to be here with you, and I'm grateful to the supporters, the sponsors, and the organizers for the opportunity to be here and speak with you, and for the logistical support to be here. This little plant behind me is dwarf hatpins. It's a G2 species, which means it's vulnerable across its entire range. And it's one of the many species we've propagated at the State Botanical Garden of Georgia for conservation purposes. What I'll be talking about today is the work that I do with many partners and collaborators, both at the Atlanta Botanical Garden and now at the State Botanical Garden of Georgia at the University of Georgia. The question we are asking is, how can we translate what we know about how plants grow to better conserve them? And this is important because all reports are suggesting that approximately 45% of our flowering plants, almost half of our flowering plants, are threatened with extinction. And it could be as high as 77% of undescribed plants may be at risk with extinction. This little trillium here, which I would say is pretty charismatic, it almost has eyelashes, was just described in 2017. So it could have been one of the undescribed species that was lost. It's found in only one population in Georgia and is now also propagated at the State Botanical Garden in a safeguarding collection. A recent report by Wes Knapp et al. Found that 65 taxa in North America have gone extinct since the mid-16th century. That's about 1.4 taxa per decade. 42 of them were endemic species, which is not surprising. And 40% of those that went extinct were from the southeastern United States. 7 of them are extinct in the wild, but still propagated in collections. For one example, Franklinia altamaha, which we're all familiar with. Another challenge that we have, though, beyond species loss, is the extinction of ecological connections. And new models show that we may be losing species and their ecosystem function before they're actually extinct. Another study found that rare species, looking at alpine plants and tropical trees, that rare species disproportionately increase the potential breadth and function of ecosystems across spatial scales, and are likely to ensure against future uncertainty arising from ecosystem change. And that there are likely to support the most vulnerable functions with no other species carrying similar combinations of traits. Another challenge we face is the loss of connections between people and plants. Plants are necessary for food security, ecosystem resilience, but they're also necessary for human health. And in areas where there's a nature gap, botanical gardens, and we're hearing from some great botanical gardens today, can help bridge that gap and create connections with plants that are important. We also need to increase data for culturally important plants. One way that we do this at the State Botanical Garden of Georgia is through field trips and educational programs, as you might imagine. This is one photo from one of our field trips. Another product we've produced is from our conservation horticulturalist, Heather Alley, who took her research on native plants that support pollinators and put it together for the public as one of the many resources we have for the public. So it includes her advice and research written for the public on 30 pollinators, 30 perennials for pollinators, not only where they grow, how they grow, who they attract, but also how to propagate them for landowners in their own backyard. So how do you propagate these plants from seed or from cuttings? North America, southeastern North America has been identified as the latest biodiversity hotspot by NAS and other authors. These maps from the Biota of North America project show areas of very, very high species diversity. Both the southeastern United States and the California floristic province both show very high densities of plant species, but they also show areas of high endemism. So a hotspot is a place with very high endemism and an area where 70% of natural habitats are threatened with extinction. One of the ways that at the State Botanical Garden we are addressing some of these challenges is through partnerships. And for nearly 30 years, the State Botanical Garden has coordinated the Georgia Plant Conservation Alliance. In these pictures are many friends and colleagues who've been working together for decades, managing habitats, propagating rare species, and even experts in their field surveying rare plants. This is a picture of Rhus machoii, which I'll talk to you about in a few minutes. And every orange flag indicates a new stem of Rhus machoii, which is quite amazing when you hear where it came from. The Georgia Plant Conservation Alliance is made up of more than 75 organizations and nearly 300 experts. Some of them are agency partners and federal agencies or state agencies that provide regulatory oversight. Others provide land management and access to rare plants, and others, like botanical gardens, are involved in research and conservation horticulture. There are over 700 imperiled species or species of conservation concern in Georgia, and from this map you can see they occur across the state. In our state alone, more than 90% of the land is privately owned. So it really depends on partnership in order to access the plants, get to find out what they need, prioritize what they need, and identify the right conservation action for them. These are some of the species that we have helped to conserve through conservation horticulture at the State Botanical Garden of Georgia and other botanical gardens in Georgia. At the heart of what we do through these partnerships is conservation horticulture. It's been there from the beginning in the early 90s when the Forest Service and the state botanist at that time, the Heritage Program, reached out to the botanical gardens and said, we only have 18 mountain purple pitcher plants left. Can you please propagate them clonally in the nurseries while we seek to find solutions for habitat restoration? So we, through conservation horticulture, are working to conserve the species in this slide and 100 others. And as conservation horticulturalists, we're also contributing to the State Wildlife Action Plan, which is currently being updated for 2025. Priority conservation actions in our State Wildlife Action Plan, which also directs where funds are allocated, include conservation horticulture, safeguarding populations, and developing a Georgia native seed network, so we have more native seed available for restoration. These are examples of some of the species that students have also worked on with us, applying technological and scientific advancements to help us understand how to propagate them. And each of these represents species that we've studied with students and with collaborators, and oftentimes students are the first author on these publications. I'm going to focus on one, Rhus machoii. So Rhus machoii is an Anacardiaceae. Dwarf sumac is its common name, and it's one of the rarest shrubs in North America. Originally, it was found from Virginia down to Florida. It's now considered extinct in South Carolina and Florida. with the largest population still occurring on DoD lands in Virginia and North Carolina. It forms clonal colonies in rocky woodlands. It's about 1 to 3 feet tall. But the most important thing to remember about dwarf sumac, besides the fact that it's a very hairy plant, is that it is dioecious. It has separate male and female plants. So in this photo here, and this is really one of my favorite plants, this is a female dwarf sumac in a conservation collection. The map on the bottom shows Georgia. And we originally were aware of there were 5 documented populations of Rhus moshoii in Georgia. But by the end of the 20th century, only 2 populations were known. The easternmost population, circled in red on the right, was all made up of all male stems. And in 2009, when I first met this species, there were only 5 stems left in the wild population. The other population that was left in Georgia is more central Georgia, very close to Atlanta, and that was an all-female population. So they were separated by 160 kilometers, which doesn't promote propagation by seed in nature. And so we set out to try to figure out what we could do for this species. So this is a picture of the male population, the eastern population in Georgia. And you can see that there's encroachment by trees. Previous research has shown that the different genders of the plant respond differently to competition, especially competition through shade. The females stop producing flowers. So in areas where fire has been excluded, you see fewer females or fewer reproductive individuals. So the plan at this population that was all male with 5 stems was to introduce fire. But again, like in other situations, we were afraid that by changing how the habitat was being managed, we might threaten this very rare shrub. So we were asked to harvest 3 of the stems, and these can propagate clonally, harvest 3 of the stems and bring them back to the nursery, bring them back to the greenhouse, and try to propagate them into more stems while we applied fire to this habitat. Over time, those 3 stems became 7 over about 2 years, so we were feeling pretty good about that. This is the female population closer to Atlanta, and there were about 150 stems there. I also want to recognize the 2 interns, Shudiyat Ajao, who went on to study environmental ecology, and later she went on to get a master's in creative writing. And then David Toms, who during this internship, while I was at the Atlanta Botanical Garden, he was finishing his undergrad at Gwinnett Technical College, and he went on to do a PhD in cellular molecular plant biology, and now he's faculty at Florida State in the biology department. So he helped with this. And what he did, actually, so this is the female population. So what we decided in 2010 was to do something of a botanical dating service. So we harvested 12 stems, or actually closer, 22 stems from the female population near Atlanta. And this was done as close as we could get to Valentine's Day. Valentine's Day happened on a weekend, so we did it that Friday. And we brought them out to the male population in the eastern part of Georgia and planted them to see what would happen. So this map from that time, or this graph from that time, shows where we're starting. So this is, again, the eastern Rhus moshoii population. And in 2009, we were starting with just 2 stems left in nature. And we applied fire in 2009. And in 2010, we introduced the female stems into the population and then monitored to see what would happen. And by the time we finished monitoring in 2014, they still conduct monitoring, but we were beginning to see exponential growth of the stems. They were beginning to take off. And so then we asked the question, are these all just those 2 stems propagating themselves in the population, or is there something else going on? So we went back to the literature, and in the early 90s, Sherman Broyles and other authors published a population genetic study of Rhus moshoii and 2 other congeners. So 9 populations were sampled, and a mean of 4.1 multi-locus genotypes per population were found. These were populations with over 200 up to 500 stems, so much larger than the population we were dealing with in Georgia, from North Carolina and Georgia. And the range of multilocus genotypes ranged in these populations from 1 to 7. So what do you think we found in Georgia? What are your thoughts? Many? Few? 2? We actually found more multilocus genotypes in our male population that nearly went extinct than had been found previously using the same technique. So we found 10 multilocus genotypes in the male population, that eastern population that was down to 2 stems. And in the female population that had 150 stems, we identified 6 multilocus genotypes. And just to test ourselves and our methods for collecting rhizomes, trying to capture diversity and take it to the male population, we captured 5 of those 6 identified multilocus genotypes when we moved them to the female population. So what this tells me is that management in place does work, right place, right plant, that plants can respond. There were definitely genotypes in the rhizomes underground, but that we could also propagate these plants vegetatively. We also were curious about how we could store seeds. Seed had been stored in seed banks, and we wanted to know how successful it was, and then how to bring them out of storage and propagate them. So we initiated some seed germination trials. Because this is a very rare plant, we first tested our techniques on a close congener, Rhus glabra, and identified that these seeds with a very hard seed coat, you can see that on the left there in a dissected seed, they take 6 hours in concentrated sulfuric acid to scarify, and then we can put them on media and grow them. The 2 resulting, so that was what we tried with the initial Rhus glabra, and then we went on to try it with Rhus machoii. So the first test was with Rhus machoii sourced from a seed bank, and out of 148 seeds that were given to us from the seed bank, 93% were empty. And 3 of these seeds germinated. Others were damaged in the scarification process or contaminated. However, when we tried to take them further, resulting seedlings did not survive. We had to keep working. In 2012, we collected seeds from that population where we had introduced the females, and we collected 114 seeds from that population where the females had been introduced into the easternmost population. 2 were not contaminated and germinated and survived in the greenhouse for 1 year, and then these 2 plants, those are the survivors, they were planted outside. So we went back and tried again. This time we tried seeds from 3 different types of collections. One was from, again, that eastern population where we had introduced the female plants, but in this case, we collected seeds from those females that were growing closest to the male plants, and then also female plants that were further away from the male plants. And then on the left side here is a picture of an XC2 conservation collection that was established at Zoo Atlanta, where both male and female Rhus machoii were planted. And so we, again, tested our techniques there. What we found is that 515 seeds from the females located closest to the males, 10% of them were filled, 11 seedlings survived in soil after a month. From 243 seeds collected from females further away from male plants, 0.4% were filled and 0 survived. And then from 2,600 seeds from the mixed XC2 collection at the Zoo Atlanta, only 0.3% were filled and 1 seedling survived. So what we're learning is that we have some techniques we can use. I didn't talk about it, but we can also put these seeds in cryopreservation in liquid nitrogen and pull them out. And in those trials, we were seeing 70% and 40% control versus cryopreserved seed germinate. So that's a good output. We can establish seedlings in greenhouse potting soil. We were not successful at in vitro shoot propagation. So we're beginning to meet the goals, but for me, the takeaway is that even in mixed gender populations, the percent seed fill is very, very low. And this is something to consider. Even when you collect the fruits with such a hard seed coat, you need to be sure if you're putting them into a seed bank. The next story I want to tell you is Eastern turkey beard. This is a plant in the Liliaceae. It is related to Western beargrass, which is a culturally important plant collected for basket weaving and sometimes is eaten. This is what Eastern turkey beard looks like in the field. You have on the left a plant that has just flowered with a flowering stem coming up, and these are plants that will die after fruiting. They can reproduce vegetatively. The photo on the right is what the habitat for the species looks like, and it's a very dry, rocky habitat, very well-drained soil, and that's going to be important. So it's a perennial evergreen herb, several years to reproduction, flowers from late May through June, fruits ripen from October to November. We knew that pollination studies were needed. No one had ever successfully propagated this plant, XC2, from seed. And so although it's common enough in the more northern states where it's found, in southern states it's becoming extremely rare. So we wanted to ask the question, how can we address the conservation needs through propagation and horticulture for this species? One of the things we learned in collecting seed from the southernmost populations of this species is that it may be a non-traditional seed for storage. So it may be a morphophysiological seed dormancy. So we had questions about whether the seed coat was preventing the embryo from germinating, or if it might be an immature embryo. So the first tests were using scarification and an acid, this time just 4 minutes. And it did seem that once the seed coat was nicked or damaged in some way, the plant could begin to germinate and the plant cells multiply, at least in vitro. We also looked at the size of the embryo through dissection and found that it did indeed look to be an immature embryo, and that once we excised the embryo and put it into the growing media, we could get the plants to grow. So our findings for this species are that it's an intermediate seed type with double dormancy, and they're hydrophilic seeds. So when they're dried down, they die. So if you dry down the seeds, they will decline, and there will be 0% germination over a year to 2. So they do not tolerate dry desiccation. If we can keep them moist in a cool temperature or in a soil bank, the seeds may remain viable for years. Through a process of alternating warm and moist cold stratification with cold to warm again, it provided the opportunity for seeds to germinate and grow. So the pictures I'm showing you are from seeds that were treated in this way. Another thing we learned, and it's sort of a basic lesson, but it's always good to be reminded of these things, is when we were able to get plants to germinate, but we put them in a conventional potting soil medium, they would die within 4 months. And again, that's just going back to the way the plants grow in nature. When we began to grow them in a very well-drained soil that mimicked the conditions in the field, we were successful. So we've been able to germinate for the first time, grow these plants to a large enough size where we can plant them back out into the field. And that's great because there's only one population left in Alabama. And so we're working with the Forest Service to put these back in. We have about 70 more to go. So the last thing I want to talk with you about is research that was funded by National Science Foundation in partnership with my co-authors, Jennifer Boyd, Will Rogers, Jill Anderson, Carol Baskoff, and Jessica Brzezinski. And with this, we asked a different question. We wanted to know, what can we learn about how plants grow to determine why some plants are rare and other plants are more common? So before going on to this next slide, this is a picture of Pityopsis ruthii. It's a very narrow endemic. It's a very pretty plant, but it grows in a very, very narrow range in the southeastern United States. So while we're asking this question, why are some plants rare and other plants more common? We were also thinking about the changing environment. And so it's helpful to think about what strategies plants have to respond to a changing environment. A plant can migrate if it's good at that. A plant can adapt. A plant can acclimate to changing conditions, or it can go extinct. We've already talked about why that's an option that we're worried about. We decided to test the levels of genetic diversity in our rare versus our common species as a potential indicator for the abilities for these two species to adapt to a changing environment. We also looked at phenotypic plasticity as a potential indicator for how these plants might acclimate to a changing environment. This is the habitat in Tennessee for Pityopsis ruthii. It exists in only the least hospitable cracks along the river. So where there can be competition for light, along the Ocoee and Hiawassee River, which are managed by the Tennessee Valley Authority, it can flood 116 times per year and then be in drought conditions during the rest of the year. So these are extremely changeable conditions. These are the two species we were looking at, Pityopsis ruthii, which is susceptible to extinction in an extremely narrow endemic, and then the more common Pityopsis graminifolia, which is considered a least conservation concern. I should mention that Pityopsis ruthii is federally endangered species and it is only found, when I say a narrow endemic, I mean it's only found along 4.6 kilometers of the Ocoee River and 5.7 kilometers of the Hiawassee River in Tennessee. So very narrow parts of the river, and with only 4 populations along the Hiawassee and 3 populations along the Ocoee. Comparing their distributions, I felt I needed to circle the dot where Pityopsis ruthii is just so it didn't get lost. So that's on the left. In Tennessee, just on the border with Georgia, and then Pityopsis graminifolia is found in a much wider range, and the dots indicate where we collected seed. Pityopsis graminifolia is common across the southeastern United States in sand hills, dry woodlands, forests, road banks, along the sides of railroads. It reproduces clonally and from seeds. There's been evidence to show that there's reproductive success with fire management. So one of the first things we learned from our experiment came well before the data collection period. And that was that Ruth's golden aster was pretty easy to grow compared to some of the other rare species we worked with. It germinated easily and grew well in the greenhouse and nursery. Pityopsis graminifolia also grew well for us, and we had over 97% survival in the growth chambers throughout the experiment. I also want to recognize our REU students who contributed to this research. Mia Rochford is finishing up her PhD. She's on the far right. And then our other students, Sophia, Annie, and Derek, all were undergrads contributing to the research. We were able to recruit Derek from the Navajo Nation, and he came to the University of Georgia to work with us and then went on to do his master's in the College of Ag. So when we tested species level genetic diversity, we just looked at overall species diversity. And there were no significant differences in the Shannon or the Simpson's diversity indices, but there was differences in NAES in terms of the numbers of alleles per locus. And there were significantly high numbers of alleles per locus for the more common Pityopsis graminifolia. This is not unexpected. This is actually what we would have expected. And then the overall MANOVA analysis of all of our measures of genetic diversity also indicated a significant difference, again, with the majority of the genetic diversity being held in the more common species. So what we did is we took seed collected from the wild, grew it up, and randomly assigned it to treatments in the growth chambers for the phenotypic plasticity portion of the project. We compared ambient conditions based on what this plants experience in the wild. We also grew plants under lower light to simulate the effect of competition. We grew plants under higher temperatures and higher water levels. And the traits we measured for the phenotypic plasticity studies included height, root mass, total mass, specific leaf area, as well as we destructively harvested at the end for root shoot ratio, root mass fraction, and specific root length. I want to acknowledge Jennifer Boyd. She led the research on the phenotypic plasticity study. So the capacity to acclimate in the face of climate change through phenotypic plasticity could be especially important for the persistence of species, especially that for species that have constrained capacity for migration and adaptation. These graphs show what potential reaction norms that we might find when we're modifying the conditions in the growth chamber. So the environmental change along the x-axis and the trait that we were measuring from that list of traits that I just showed you against those two environmental differences. So if there's no plasticity, we don't expect to see any change in the reaction norms. If there's some plasticity, we might document those changes over a changing environment or see very highly variable, strong plasticity, evidence for plasticity or genotype by environment interaction. This is my chance just to take a breath and show you again the students working in the greenhouse propagating these plants. And I'll say this, although Pityopsis ruthii is extremely rare in the wild, it would be a great horticultural plant. It's quite beautiful with silvery leaves and it grows well in cultivation. So these students are making crosses with the plants. So to test phenotypic plasticity, we did two-way ANOVAs, both species by abiotic factor and population by abiotic factor. And for significance, we used RDPI, plasticity index analyses. I'm just going to focus on the species by abiotic factor for the interest of time. So in response to light, interactions between the species and abiotic factor were significant in all light, temperature, and water experiments for multiple traits. And here the reaction norms are graphed. In response to light, there were significant differences in phenotypic plasticity between Pityopsis ruthii and Graminifolia. Specifically, Pityopsis ruthii showed more plasticity in these traits than the more common congener did. In response to temperature, the reverse was true. Pityopsis graminifolia showed a stronger plasticity with changing temperature than Pityopsis ruthii did. And finally, in response to changes in water, increases in water, Pityopsis ruthii showed more evidence for significant plasticity in changing environment compared to Graminifolia. So it's been proposed that general species occurring across a broad range of habitats may be characterized by high plasticity relative to species with narrow habitat specificity. But our findings indicate instead that plasticity could be facilitating or resulting from the specialization in a very narrow habitat with very significantly changing habitat conditions over time. Specifically, we found that the rare endemic Pityopsis ruthii exhibited higher plasticity in response to both altered light and water availability than its widespread congener, Pityopsis graminifolia. And although contrary to expected differences, these findings could help to explain the specific habitat restriction of Pityopsis ruthii to the boulders along two rivers with dynamic water flow and the persistence of plants in locations ranging from sunny to fully shaded. We also found that Pityopsis ruthii did not exhibit plasticity in response to temperature, which contrasted with its common congener. We suggest that an inability to acclimate to temperature could help explain the limited latitudinal and elevational extent of Pityopsis ruthii. So, and however, factors such as the east-west orientation, so the Hiawassee and Ocoee rivers are shown in the map here of Tennessee, North Carolina, and Georgia coming together in a very narrow range. very mountainous, very difficult to perhaps navigate those changes in temperature that would allow it to migrate out. So I know I'm out of time, but I just want to acknowledge the collaborators with many of these projects, particularly Will Rogers, who's been a friend and a collaborator over many, many years, and he contributed significantly to all of these projects. Jerry Pullman, he's now retired from Georgia Tech, but he led the research in vitro and for cryopreservation. Kylie Bucalo worked on these projects both at Georgia Tech and at Atlanta Botanical Garden. And I acknowledge my co-PIs and collaborators on the NSF project, as well as Michael Bonzel and Jeff Call and Adam Datillo from U.S. Fish and Wildlife Service and Tennessee Valley Authority. So thank you. I appreciate your attention. Okay, great.
Don't be shy.
Yeah, we have one question online, and I also have my own personal question.
Okay.
So question online, actually, a bunch of questions are pouring in.
Oh my.
One question online was, does the State Botanical Garden of Georgia produce an index seminum, which is seed catalog?
We do not produce a seed catalog. We collaborate with Botanic Garden Conservation International. We use IRIS-BG to document our collections. And we're now launching a Georgia Native Seed Network where we'll be collecting native seeds from across, and that's a whole nother fun story I could tell you. But no, we don't produce an index seminum.
One of my own questions.
Okay.
So we heard three really interesting stories here.
Yes.
What criteria do you use to decide what plants to grow in the greenhouse? Is it based upon just pure love of a certain individual, or is it based upon how rare it is?
Sometimes it's love. Yes, we do fall in love with certain species. It's guided by the Georgia Plant Conservation Alliance, that alliance of experts from across the state, from regulatory agencies who give us permission to work with these species, from the land managers or owners, and from our own knowledge. So with all of that expertise, we identify the top conservation priorities in our state, and that's where we like to focus our efforts. That way we know we're contributing to the most pressing conservation needs.
Two questions, if I may. One, does the state of Georgia Botanical Garden participate in the National Seeds of Success program?
That's a great
And if
question.
so, how?
Okay, thank you for asking that question. We do. We are beginning to participate in the Seeds of Success program, primarily through the Southeastern Grasslands Institute and our partnership with SGI. So by we're identifying Piedmont prairie and grassland species as being a top conservation priority for Georgia and for the State Botanical Garden. So we just had a training workshop for SOS at the State Botanical Garden, so we could both train our staff and collaborators from surrounding areas in the methods of SOS. We sent our first seed collections last fall, and we'll continue to grow our participation in that program. Thank you.
Thank you. And the other one is about the sumac. I did some coordination on this with the military in Virginia, and up there they say that there was the dwarf was cross-pollinating with other species, and so maybe you could speak to that a little bit and see if you had that issue in Georgia.
That's correct. There is some indication that there could be hybridization between the very rare Rhus machoii and some of the more common Rhuses that co-occur. That's an area that we're actively researching right now. So we've been able to produce seeds in mixed collections at the State Botanical Garden, and we're beginning to ask those questions. I've been wondering if the very, very low seed fill rate in fruits that are collected for seed banks from Rhus machoii may represent hybridization events. I'm not sure. So that's something we really need to be looking into. Thank you. One more. Okay, thank you.
Okay. I actually had two questions. They're fairly quick. One is you mentioned that 65 taxa had gone extinct since European settlement, and I was wondering if you could give an indication of sort of what percentage of existing taxa in North America that was. And the second was relating to the potting soil that you were talking about for the plants that you collected from the site. And I was wondering if you could talk a little bit about the characteristics of the soil to reproduce the potting soil, or if you just collected soil directly from the site, or just a little more detail on that.
Those are
Thanks so
great
much.
questions. Thank you. This research you're referring to was by Wes Knapp and other co-authors, and it was produced in 2021. So I can provide you with that citation and the research they used to identify. That was not our work, but I'm referencing that study. And Wes Knapp is the botanist at NatureServe. So they were looking at the distribution and known extent records for native species in North America. Wes Knapp 2021. In terms of the soil mixtures, the first one was just just a traditional greenhouse mix that held a lot of water, because usually what happens with, I'm looking for my notes to see if I can find if I had more in my notes about that, but it has vermiculite. It was not at all based on the natural characteristics of where the Eastern turkey beard grows. So it held a lot of water. So it was your basic nursery soil, vermiculite, a really spongy soil. We switched that out with a soil that had more sand in it, had no vermiculite, and had aged pine bark in it. And that one was far more successful. We also hypothesized that the pine bark in the soil, so it was a very well-drained soil, the one that was successful. And we also hypothesized that the aged pine bark brought microorganisms in that may have helped the roots be healthy in cultivation. And again, I can share that citation with you. Happy to.